expression. So the circadian clock, and at least the timing of specific flower events, has been known for a long, long time. And even Linnaeus noted how precisely the timing of opening of different flowers is, and he proposed that you could plant a garden of different flowers and tell time simply by taking a stroll through your garden and looking to see which flowers are open. Since then, we found how the circadian clock regulates flowering time and when to flower in plants, as well as volatile emissions, which help to coordinate plant-pollinator interactions. So in the Hormonal lab, where I did my postdoc, we found that juvenile sunflowers, my colleague Hagar Atamian did a very nice study looking at how the circadian clock regulates heliotropism in juvenile sunflowers. So heliotropism in juvenile sunflowers is the process where they slowly track the sun from east to west across the sky during the day. And one of the first clues that this might have been regulated by the clock was the fact that at night, in the absence of any external cues, these plants reorientate back to the east in anticipation of the following day. And this only happens in juvenile sunflowers. Once sunflowers mature and start to bloom, they all maintain a similar orientation and usually face east. If you've ever taken a stroll through a sunflower field at dusk, you'll see how they usually face away from the setting sun in the west. So this led to a question about how do environmental cues and circadian clocks regulate head orientation. So part of the study, Hagar noted that as a sunflower matures, it seems to maintain a robust tracking to the east every day. But as it gets older, the tracking to the west appears to decrease over time until it's maintained facing east. And so he did a neat little study where he placed juvenile plants in front of a light at different times of day. And one of the main findings from this was that the plants seem to curve more strongly towards the light in the early hours of the day compared to the afternoon. So this suggests that the clock was gating the plant's stem response to light to the morning hours. And so this is likely what was facilitating that robust tracking to the east as the plants grew, and probably helped maintain the easterly orientation once they stopped tracking. So this study really picks up from there, where the next question was, well, is there a biological relevance or importance to the orientation of a sunflower? So why do they face east? To answer that question, we planted sunflowers in pots in the field, and once they had completed their tracking, we turned half of them to face west. We then measured pollinators counts by video analysis. We did time-lapse photography of floret maturation. We also dissected florets to measure floral organs and manipulated temperature by heating capitula. And then we monitored temperature through all of the experiments. And one of the first things we found, especially along with our collaborators at the Blackman lab, was that east-facing plants tended to have more earlier insect visits than west-facing plants represented in the blue here. When we looked at the temperature data, we actually saw that east-facing plants heated up and were hotter earlier in the day compared to west-facing plants. This really makes sense because they're facing the rising sun. So we then took a west-facing plant and artificially heated it to mimic the temperature changes we observed on east-facing plants. And then again counted pollinator visits. And we could see that the artificially heated west-facing plant, represented here in orange, showed an increase in insect visits. And this suggests that temperature of the capitulum is coordinating pollinator visits in some way. So the next question we had was if these higher early morning temperatures could actually be affecting the flower and seed physiology and pollination, since there seems to be a link with pollinator visits. So the first thing we looked at was seed traits, and a very talented undergraduate assistant I had at UC Davis, Sana Saeed, took a number of different measurements from different trials over several seasons. And we looked at capitulum diameter, dehulled seed width, and seed weight. And in each case, she found that the east-facing plants, represented in red, had higher values than the west-facing plants. And so this suggests that there might be a female fitness advantage to facing east. But we did see that this was locality specific, and we didn't see the same trends when we looked at data collected from the field site at Charlottesville in Virginia. I'm not going to go into much details about this, but I'm happy to chat about the possible whys after the session. So then we wondered if there was also a male fitness advantage. Did it affect pollination? And this study was, or this experiment was designed, very elegantly designed by members of the Blackman lab, where they took cytoplasmic male sterile plants, so these are plants that do not produce pollen, and put them in the center of the site. They then had known sires with known molecular markers that they placed either facing east or facing west, and they switched the sires from east to west and also switched in and out different sires. And allowed pollination to occur. They then harvested the seed, germinated it, and did paternity tests to assess which sires produced which offspring. And from this, they could see that in almost all cases, the east-facing plants produced more offspring than the west-facing plants, suggesting there is a strong male fitness advantage to facing east. So we wondered if this increased pollination could be due to a different timing in pollen presentation. So we went back to our field and looked at our east, west, and west heated plants. We imaged them every 15 minutes. And then we went to each image and counted the number of florets that had pollen across the time series. And from this quantification, we could see that the east and west heated plants, represented in red and orange, had an earlier timing of pollen presentation when we compared it to the west-facing plants, represented in blue. So then we wondered, well, how does temperature regulate the timing of pollen emergence? And for this, we needed to think a little bit about floret physiology. So in a sunflower floret, you have the corolla that houses the anthers cylinder. These are 5 fused anthers that form a cylinder, and they sit on top of anthofilaments. Through the center of this, you'll find the style and the stigma. When the corolla opens, the anthofilaments start to elongate and push the anther cylinder out the top of the corolla. The pollen then is released into the center of the cylinder, and slowly the style and stigma push through the center of the cylinder, pushing the pollen out the top in a plunger type mechanism. And then only the next day does the stigma unfurl and is receptive to pollen. And we wondered how temperature might be affecting this process. So back to the field we went, and we looked at our east, west, and west heated plants again, and we measured the length of styles every 15 minutes across these different plants. And what we saw was that the styles of east and west facing plants appeared to elongate much more rapidly than the west facing plants over time. We also looked at the length of the anther and as it emerged out of the corolla as a proxy for anthofilament elongation, and we could not see a difference in these plants for anthofilament elongation. Other studies have shown that anthofilament elongation appears to be very light sensitive, and so it's interesting to note that style elongation and anthofilament elongation seem to be differentially regulated by different environmental cues. To confirm the temperature changes that we have temperature regulation, we did a chamber experiment where we heated a subset of plants and kept all other conditions the same. And again, we saw the heated plants had an increased style elongation and earlier pollen presentation, linking temperature regulation to style elongation and pollen presentation. And that's really where the study ends. And then I relocated to University of Pretoria for my faculty position, and we followed up on this temperature link. So my MSc student, Uyamamela, who just submitted a dissertation, did a very nice study where all her plants faced east in the field, and then she just heated a plant to 10 degrees above the natural temperature of the plants. And we assessed style elongation, pollen presentation, and insect visits. And we could see that, again, increased style elongation in earlier pollen presentation. Surprisingly, we didn't see a change in insect visits, but this is likely because insect visits are coordinated not only by temperature, but also by light and several other factors. We were lucky enough during this time to have a natural heat wave in the field, and so we also assessed natural heat wave effects on these processes. Unfortunately, this block is grayed out because it was a casualty of COVID, and due to curfew regulations, Uya couldn't get in the field early enough to actually see the changes of style elongation. But we did manage to see changes in pollen presentation under a heat wave. It also shifts earlier. And under the heat wave, we also saw an increase in insect visits to an earlier time point, suggesting that there might also be a mechanism to ensure pollination earlier under heat stress events to help limit damage. So overall conclusions are that sunflower orientation affects plant fitness and coordination of pollinator visits. Temperature regulation of style elongation coordinates the timing of pollen emergence. And our newest results suggest that style responsiveness to temperature may be a mechanism to limit pollen exposure to high temperatures later in the day under conditions such as heat waves. And with that, there's always many people to thank with this kind of research, especially everyone at the Hormonal lab for hosting me during my postdoc, awesome collaborators at Ben Blackman's lab, and then my wonderful cohort of graduate students and postdocs that I work with every day now. And thank you for your time.
Wonderful. Thank you very much, Nikki. So I'm just going to jump in and ask one quick question. I've always been fascinated by secondary pollen presentation in Asteraceae. It's so cool, and all the different mechanisms that they have. So does that itself also have a circadian pattern? You were talking about how they tend to push it out early in the day?
So we're not sure. We think so. That's actually, I left off when I left the Hormonal lab, I left off that experiment, and another postdoc, Corinne Marshall's taken over those experiments. So she's done a bunch of chamber experiments with different light conditions, constant light, constant dark, a number of shifted T-cycle type experiments. And I think she's busy preparing that paper now. So there does appear to be a link, but it's still to come. Watch this space.
Right. And I'm seeing a lot of questions pop up, and I don't know if we'll have time to answer all of them, but I'll start with Eyal's question. Have you checked somehow to see if the circadian clock rhythms are themselves changing under higher temperatures? So how is maybe even the diurnal variation in temperature influencing the clock itself?
We haven't done many temperature experiments. I think Corinne added that to hers as well. So my study was more just understanding the temperature relationship with pollen at that stage. But yes, that would be an excellent avenue to check as well.
I think we're gonna have to save the rest of our questions. I know I have to go on to the other. There are a bunch of questions. We'll have time to chat after the presentation, so I'm hoping we can get to some more of these questions because there are still a lot of really good questions. But we're gonna go on to our next speaker. But thank you very much, Nikki. Our next speaker is Luisa Lenay de Barros Dantas, who's a research assistant at the John Innes Center. And she's going to be telling us about some work they've done to look at how field microenvironments regulate crop dial transcripts and metabolite rhythms. So thank you very much, Luisa.
Right, I'm all set now. Hi, everyone. Let's talk a little bit about field microenvironment and high density crops. I'd like to highlight first that I am co-first in this publication together with Mayra. Mayra is a talented, brilliant master's student at Carlos Rota's lab. I think she's about to finish her master's and so she'll be ready to start her PhD. And I would like just to point out the obvious for most of us here, that the circadian oscillator, it regulates either directly or indirectly a plethora of plant phenomena, important plant phenomena related to physiology and metabolism. I mean, from flowering, as Nikki has shown in her amazing presentation, all the way to growth, to primary metabolism, to photosynthesis. The circadian oscillator has a degree of regulation in them all. And this was established in experiments that were done under controlled conditions. And that's how they were established inside of growth chambers with Arabidopsis, our model, where they were even smaller, still at seedling stage, and not always in soil, sometimes in MS plates. So as we move forward as a community, we started to wonder, what about plants in their naturally fluctuating environment? What's the extent of the circadian regulation in such fluctuating environments? And we had some studies starting to shed a light on it. First of all, we had a study showing that this mutant in an important circadian oscillator gene completely lost its phenotype once it was growing under natural field conditions. We also know that chambers sort of fail to fully mimic the quality and spectrum of sunlight. And we also know we have strong evidence proving that the same Arabidopsis line grow in other controlled conditions, in phytotrons or growth chambers, and in fluctuating environments, they show completely different metabolic profiles. So what we've done here was to grow a high density crop. The model that we used was sugarcane, and we grew this high density crop in a field, and we analyzed the plants when they were in two different developmental stages, when they were still young, 4 months old, and they were older when they were 9 months old. And this is what we found, because we're lovers of the circadian oscillator. We looked first at two homologs for LHY and TOP1 genes transcripts. And what we saw first was that when we compared the transcript profiles of 4 months old plants and 9 months old plants, we observed that the plants, when they were 4 months old, LHY peaked 0.2 hours after dawn, whereas when the plants were older at this stage, the transcript LHY peaked 2.7 hours after dawn. So it was obvious here for us that LHY, this transcript, had in 9 months old plants sort of showed a phase shift compared to 4 months old plants. So we decided to go further and look at the transcriptome scale within this comparison. And at first we noticed that the level of expressed transcripts was higher in 9 months old plants. So the 4 months old, the number of expressed transcripts in 4 months old plants corresponded to only 86% of the ones in 9 months old. And this sort of pattern also was found for the rhythmic transcripts. Around half of the expressed transcripts were rhythmic in 4 months old plants, whereas 68% of them were rhythmic in 9 months old plants, which means that all the plants had a higher level of expressed and rhythmic transcripts. And not only that, what also happened is that the plants, when they were older, the transcripts started to there was a phase shift to be expressed and peaked later. And just look at the gene ontology and just break down the categories of those transcripts, we could also find some interesting things. First of all, what we found is that there were some categories of transcripts that showed a high proportion of rhythmic transcripts in both of the 4 months old and 9 months old plants. either for circadian clock or transport categories. We also saw some categories that had a higher proportion of rhythmic transcripts in 4-month-old plants, like ethylene signaling, or in 9-month-old plants, like protein synthesis. And what we could consider is that the proportion of rhythmic transcripts in different functions, they sort of changed to in 4-month-old plants and 9-month-old plants. And then when we had this entire scope in data for transcripts, we decided to look at metabolites and also at flowering genes. Before moving to metabolites, I would like to say that flowering genes, they also showed a phase shift in important genes, homologs found in sugarcane associated to flowering. We saw that flower locus G was only expressed when the plants were 9 months old, which is quite understandable considering that the developmental stage of the plants were in 4 months old compared to 9 months old. And we do see this pattern in other grasses for FFD homologs. Whereas for other genes associated to flowering sugarcane, what we do see is either a 2-hour or a more than 2-hour phase shift for later peak expression in those genes. And as a takeout message, it's just that genes associated with flowering also follow the trend that we've seen for LHY here, with a transcription pattern for later transcription in 4-month-old plants compared to 9-month-old plants compared to 4-month-old plants. And we went now look at metabolites, arrhythmic metabolites, and we first saw that the trend of higher percentage of rhythmic metabolites in other plants was also confirmed, as we saw a higher percentage of rhythmic transcripts in other plants as well. And when we performed the principal component analysis in 4-month-old plants or in 9-month-old plants, we saw that night and day time points were clustering apart for a couple of time points associated with early, beginning of the day, or to the end of the night. But in general, we did see a phase shift, as you can see in panel B. And just breaking down the phase shift categories of those metabolites, we could see 4 different categories. We saw 2 metabolites that peaked earlier. This is sucrose, and if I'm not mistaken, myo-inositol. Then we saw 3 metabolites that peaked within 1 hour apart when we compare 4-month-old and 9-month-old plants. Then we saw a 3rd category of 10 metabolites that had a phase shift of 1.5 to 6 hours, with again 9-month-old plants peaking later. And we saw 5 metabolites with an even broader phase delay of 8 to 13 hours, with the older plants also peaking later. So the trend continues as we looked into metabolites. And then we started to think about what was going on with those plants. And this is the hypothesis that we came up with, the sort of explanation that we came up with that gave sort of, that was important for our hypothesis. So what we thought was happening was that the plants, when they were 4 months old, they were still very small and they were not shading each other in the field. That means that the sun was capturing and the plants in a uniform way, was reaching in a uniform way the plants in the field. Whereas when the plants were older, they started to create a dense population in the field by mutually shading each other with their senescent leaves and with their still photosynthetically active leaves. So we started to think, what if the phase shift that we see here in LHY, in flowering genes, in rhythmic metabolites is driven by a local microenvironment that is caused by mutual shading. And then we proposed 2 experiments to test this hypothesis. We've at first decided to harvest plants just from the east and west side of our field. And we proposed a second experimental approach in which we built a wooden barrier, and we harvested the plants just before the barrier and just after the barrier. And our goal was to investigate how was the LHY transcript expression to use as a proxy for our hypothesis. Because let's not forget that LHY of all the circadian oscillator genes is the one that is more sensitive to light. It is a sort of a proxy as well for dawn, for sunlight perceived, for the sunlight that is perceived by plants in a field. So it was really interesting to see when we looked at the transcript profiling of the plants that were harvested from the east margin at dawn and west margin also at dawn, to see that we saw again that the east margin plants, the plants that got the sun first, they had a peak of expression of LHY around 0.6 hours after dawn, whereas the west margin plants that got the perceived sunlight later had also a LHY expression peak around 1 hour later. And the conclusion that the takeaway message that we got from this experiment is that the plant location within a field sort of was important for the dawn perception by the oscillator, because again, we saw the LHY shift, but as we've seen before, even in the 4-month-old versus 9-month-old comparisons, nothing happened in terms of phase shift in the transcript expression of top one. And in our second experimental approach, when we were analyzing the plants just before our wooden barrier, our wooden wall, and the plants just after the barrier, both of them, when they were harvested at dawn, what we saw again was the same pattern in which the plants before the wall, they had LHY peak expression 1 hour around after the astronomical dawn, whereas the plants after the wall, they had a peak of expression LHY around 2.5 hours after dawn. And so what we saw here again, it was that the plants that were getting the sunlight at dawn later compared to the plants before the wall, they also showed a phase shift in the LHY transcript expression. And this was interesting as well, because it sort of also gives us idea that LHY was a good proxy for the dawn perception by the circadian oscillator. And this was important, and I think that the most important and the takeaway message of the entire publication is that the plants, they are in the same field. They are experiencing locally microenvironments that might come for our model of a high density crop from mutual shading for different temperature hotspots or different light hotspots. But the point is, this affects the circadian oscillator, and it affects how the plants are perceiving time. And ultimately, it affects metabolites, at least the rhythmic metabolites. And for crops, this has an impact, a direct impact on yield, and as we saw, on flowering time. And those are all important traits that we are looking for in agronomical important plants. And this work was also graced the cover of the Phytologist last November, which we're very happy about. I was happy because I took the picture. And I think that Carlos was happy because it's now a cover board right here. And with that, I would like to just acknowledge everyone that got involved in this publication. I was supervised by Carlos, who's the main author in our paper. I had fun with my PhD. He was a great mentor, a great supervisor. I would also like to highlight Mayra, the co-first author in this publication, and our collaborators, all of them Brazilian. This is a Brazilian, 100% Brazilian science funded by a Brazilian grant agency. This means a lot to us. And I would also like to highlight and acknowledge my former postdoc supervisor at the Max Planck Institute and my current line manager at my side house, the Joinas, for their support during the final stages of the manuscript preparation. And that would be all.
Thank you so much, thank you so much, Louisa. That was a wonderful talk. Unfortunately, I think we need to go on to speaker 3, but we will have a significant Q&A session at the end. And we've got all the questions lined up here that we'll be able to take at the end. So thank you so much. So we now have our third speaker, Manas Prosti, who is a postdoctoral fellow at the Institute of Plant Sciences at the Agricultural Research Organization of the Volcani Center. So Manas. Thank you so much.
Hi. Thanks, Elena, for introducing me to this event. I feel privileged to be presenting in this platform. Today, I'm going to speak about our work on the circadian clock plasticity. Hello, Elena. Is my presentation is visible too?
Yeah. You're all good.
Yeah. Okay. Yeah. Okay. So, yes. So today I'm going to speak about the circadian clock plasticity and crop productivity under barley domestication. I work in Dr. Friedman's lab in Institute of Applied Science in Air, Israel. Here we work on the circadian clock. As everybody already explained about what is this circadian clock, I would not invest some time in explaining that one. In brief, there is an input, there is a core clock, which runs is a feedback loop, and it controls many growth and developmental process in the plant. So why in our lab we look for the plastic trait for this circadian clock? Because we know since the global temperature and the global environment is fluctuating, so if we can find some of the loci that brings plasticity to the circadian clock, it will be helpful for the plants to adapt to this unfavorable condition, and it will have a high adaptive value for the this changing climate. So in our lab, especially, we work on barley, and in order to see the plasticity of the circadian clock under barley domestication, we used 3 different panels to see there and compare the change in the plasticity. So the first one is the wild barley, which is the B1K denotes here. It's collected from all throughout the Israel in 5 microsites, and this is around 360 some accessions we have. And then we have also the cultivated panel, which is denoted here as S2MET. This consists of the Hordium vulgare, the cultivated breeding lines of barley. And this is from the US spring, it's a 2-row, and it's from the multi-environmental trials panel. In addition to that, also we have a barley core collection from Germany, which also contains the cultivated variety and also the landrace. So in summary, in total, we have wild accessions of the barley, landraces, and cultivated variety. And we wanted to know what is the plasticity exists in these 3 different panels of the barley. So phenotyping of the circadian clock is really laborious, and it is there are many different ways we can do that one by molecular assay, but it's time consuming, and also it's destructive. So in non-destructive way, we have in our lab we use the SensiPAM, which is based on the chlorophyll fluorescence. So we grow all the plants from the different panels. We do the entrainment for the short day or long day, and then we measure the clock under the constant light for 3 days, and we take the data on every 2.5 hours. And we do the experiment since for the thermal plasticity, we do the same set experiment on 22 degrees centigrade, which is the optimal temperature, and on 32 degree centigrade for the high temperature. This is our platform where we use this. This is a automated phenotyping platform where we grow the plants, we put them on the machines, we tag them. In one set, we can run at least 240 lines. So here we can see you have 3 different parts: dark adaptation chamber, and then imaging chamber, and then post-imaging chamber. And when the plants go to the enter to the imaging chamber, the chlorophyll fluorescence data is recorded, and from this data we see the rhythmicity, and using some platform we deduce the period and amplitude of that particular early accession. This is how we select the area of interest. We do this experiment in much replicates so that we have a statistical power. So we did this same thing for the phenotype for barley wild, for the barley land races, and the cultivated variety. So in the initial data we see it here first. When we deduce the period, the circadian clock traits, which is period and amplitude, we compare between the land race, between the wild land races and cultivated variety. At first, if you look at the period, we see the period when there is a transition in the temperature. In the period, in most of the accession, there is a shortening of the period, and which we call as a deceleration. So, but this deceleration was mostly pronounced for the wild B1K accession and compared to the cultivated one. And also for the amplitude, if you see for the wild accession, there is a significant increase in the amplitude. Just, I think it's nearby double the amplitude under high temperature. But unlike to the cultivated line, where in the beginning the amplitude was higher than that of the wild accession, but the increase was not that much, not that much like to that wild one. If we consider for the land races, also we see the trend of deceleration of the period goes in direction of the wild panels. However, in amplitude it go in opposite direction of the wild accession. So in total, if you see the plasticity here, we see from the phenotypic data of the wild and land races and the cultivated panel, we see the wild barley accessions to be more plastic and compared to the cultivated and the land race accession. So if we see, we can say other way, the circadian clock of the cultivated germplasm looks to be more robust than that of the other two, land races and wild. So this was the previous slide, was all about the phenotypic data that we got from the machine, from the chlorophyll fluorescence. But there is also a core circadian clock in every organism. So in order to know also what is happening inside the core clock chain, so what we did, we also carried transcriptomics analysis of the different panels, and we filtered out all the core clock genes, and also some of the output genes, around 20 output genes, those who has functions in photosynthesis, light harvesting complex, like that. So we try to see what is happening in the core clock versus to the output plot, because we see a phenotype of the plasticity in the wild versus this one. So here, when we see to the core clock genes, we didn't see or we didn't observe any significant difference. So this is this data is the statistical data of all the core circadian clock genes in the barley. So here we don't see either for period or for amplitude, no difference in the core clock gene between optimal temperature and high temperature. But in other words, when we see the output gene, we didn't see any difference for the amplitude, but we do see a deceleration of the output genes for period in under high temperature. So this means the core clock is really robust, and it has a temperature compensation mechanism with transitioning the temperature from 22 to 32 really doesn't affect the clock much, the core machinery, but whereas the output activity and the output genes, they really behaved, they really accelerated with the period as shown in the phenotype. So in order to know what is the genetic basis for this, what is the genetic basis which is responsible for this plasticity? So we took another population which is called haloexotic barley Hebe population, which is a NAM population, and this is a nested association mapping population. And we took this population where we can find the intermixing of the wild allele versus the cultivated allele. So we can pronounce. So if you go for a genome-wide association study, maybe we can see the effect of wild allele versus the cultivated allele. So this population was helpful us to see and to identify the genetic loci in the genome that play or that drives this plasticity. So so we took this population the similar way we grow the plants. We took to the machine, we get the data, and we have the genotype data for this population. Then we made an genome-wide association studies. We conducted the genome-wide association study in different approach, in different ways, to make sure that the data we get is we have some consistent QTL in all of the methods. And finally, we found these loci, which we called DOC, which is also we named drivers of the clock. And for different temperature here, this is the circus, which is for period and amplitude. And this is the annotation. The outer circle is the source, the Manhattan clock, or the significant loci appear under high temperature. The next one is for the optimal temperature. And then also we did some kind of footprint of selection assay to see if there is a selection in this loci. Also, we did this selection in two different ways. One is by the CLR-based method, and another is a reduction in nucleotide diversity. So, especially we see for the period, we see one of the loci on chromosome 1, which is we named as DOC1. It appears under high temperature and for the period. And for DOC5, and for the DOC5, we found this QTL under optimal temperature. And what it does, if you see the bar diagram, the diagram below, the wild allele here in the DOC11 reduces the period or accelerate or shorten the period, and also the wild allele in optimal temperature for the DOC51 also reduces the period. For amplitude, we found DOC 3.1 and 3.2. 3.1 because they are present in chromosome number 3, and 3.1 and 3.2 because they are two QTLs and very close by. And what these two QTL does, it increases the amplitude under high temperature. And if you see their allelic effect, we see the wild allele increase the temperature here. So from this genome scan, we see we found some QTLs that exactly behave to our phenotype. We see in the wild the decrease in period. What we observe here, if there is a decrease in the period, it's because of the wild allele, and if there is an increase in period, increase in amplitude, it's also because of the wild allele. So since we see and we see in the first diagram that this plasticity is reduced when we go from the wild to landrace and landrace to cultivated gene pool. So we suspect maybe there is a footprint of selection or signature of selection in this loci. So for that reason, we took 500 accession from each wild panel and landraces, and also cultivated from a published paper because they have the genome information there. And we wanted to see if there is a selection of signature in this loci. To our surprise, we see the selection of signature on these 3 drivers of the clock loci. So it means this loci has been lost out. This loci which play for the role of the clock plasticity has been diluted to a greater extent in the cultivated gene pool. So, and also we wanted to see if there is a day length dependency of this clock plasticity for this effect of the day length dependency. So few of the accession of the hip population also we took, which is segregate for DOC31 loci and DOC51 loci, and we carried this experiment in both way. So here the first one is for the DOC31 and this is for the DOC51, and here we do for the high temperature and long day, high temperature and short day, optimal temperature long day, and optimal temperature short day. And similarly we do it for the DOC51. We see really there is a temperature, there is a day length dependency, because as you see here, the period shortening happens only during high temperature and long day. And also, if you see in the DOC51 also, under optimal temperature and long day, we could see the period decreasing is more pronounced compared to the short day. So this is all about our the loci that we found for the clock traits. But we wanted to see if this clock trait, I mean the loci which is controlling this trait, does they have any kind of pleiotropy, or they control any of the plant productive traits. So we have some data earlier for the drought tolerant, and also we conducted an experiment in optimal temperature and high temperature, and we take data like spike fertility, plant biomass, and yield under drought and high temperature. So, and yes, we see that's the loci which we named as dogs, or the drivers of the clock. We see they are also controlling the yield and also plant biomass. But the wild allele in case of the drought experiment, here is the DOC 3.1. It decreases the grain yield in drought. I mean, although this wild allele brought plasticity to the plant, but it go against the yield. I mean, the yield we get for this wild allele decreases. But if we see for the plant biomass, we see the wild allele increases the plant biomass under high temperature. So we do see this DOC loci has really pleiotropic with the crop biomass and also yield. So this is our summary of our study. So the summary is we see a loss of thermal plasticity, and this loss of thermal plasticity is for the clock output genes, not for the core clock gene. So there is temperature compensation mechanism is maintained for the core clock gene, while this plasticity is mostly for the clock output genes. And we identified a few major DOCs, which is called drivers of the clock loci, which underlie these differences. And we do see the pleiotropic effect of these DOCs on grain yield and biomass. And we see the day length dependency of the QTL, day length dependency of the QTL, and this match with the evolution of the crop, because when there is a domestication, the wild ancestor was used to grow in a short day condition, and later it went to the northern hemisphere where there is a high day length. So it goes with that theory. So at the end, I would like to thank all of my friends, mostly to Eyal Dolak, who is also a co-author in this paper, and share authorship, and also A.G. Yamamoto, and to my PI, Eyal Friedman, and all other people mentioned in this. Thank you. That's all.
Wonderful. Thank you so much.
Yeah.
So now the good news is that we have some more time to catch up on our questions. So I think they will, if you want to stop sharing your screen, we can go back to our panel and catch up on some of our questions, because there's all kinds of good questions here. I was actually going to suggest that we go back. I'm not sure. Christine, do you have a preference on how we do this? We could go back. I know Louisa didn't get a chance to answer any questions. We could go back to a couple questions for Louisa, and then we can just kind of do a little round robin here. I'm not sure if
we're happy to do that. So please feel free to go to Louisa first if that's what you want. It's your show.
Okay, great. So we'll just sort of take turns here. So Louisa, there's a question from Sandy Kerbler who asks, can you hypothesize why the number of expressed transcripts is lower in 4-month-old plants compared to 9-month-old plants? Is there something about, their growth phase, what being more mature, being immature? Yeah, what do you think might be going on there?
Thanks for the question, Sandy. And I think it definitely has something to, two hypotheses come to mind. One of the hypotheses is about the developmental stage per se, and the fact that in the leaf blades of grasses and sugarcane as well, when the plants are, and although we harvest the same leaf, they were obviously in different developmental stages. What happens is that development-wise, the leaf blades of grasses, they elongate rather than multiply, really divide in early stages compared to when the plants are older. So this is one way to look at it. The other way to look at it is that the more mature plants, for instance, plants that are 9 months older, they also have different tissues. They are still not fully developed in 4-month-old plants. That also has a role to play together with the elongation versus number in trying to tackle why 9-month-old plants have a higher number of expressed and therefore rhythmic genes.
Terrific. So please note that Christine has noted in the chat that you can also feel free to raise your hand if you just want to ask a question directly, but I'm happy to keep reading. I don't see any questions yet from Manas, but maybe I'll go back. We have some way back at the beginning for Nikki. How do you think floral temperature effects might be relevant for night flowering species?
Yeah, so that's a really interesting question, and I haven't actually spent that much time thinking about nighttime temperatures at this point. But I do know that some insects like bumblebees will overnight in flowers for warmth. So I think that does affect pollination from that aspect.
Right.
And I've also seen studies that suggest that high nighttime temperatures, so thinking climate change and things,
right,
can be really detrimental to pollen as it develops. And so those are the two things I can think of off the top of my head, but it's definitely something I want to read more about after that question.
Fantastic. So actually, I'll have a question for Manas. Have you thought about, so you've shown that some of these loci that are involved are actually under selection. Do you want to maybe expand a bit on like why you think they were under selection, or alternatively, whether you think that there might be other targets for selection in terms of sort of crop improvement in this system?
Yeah. Thank you for this question. Yeah, when we run for the signature of selection, we do see the signature of selection in other loci also. But we emphasize here, we got many other loci which has underwent selection through this domestication. But we emphasize here our dock loci. If these 3 of the dock loci that we found in this, in our analysis, if they are coming under this, if they fall under the selection of signature, so that's why we show only we show our desired docks. They are also coming under the selection. But apart from that docks, also we have many other loci that we encountered. They have undergone selection.
Sure, terrific. So I have another question for Louisa. So what kind of physiological changes would the anticipation of LHY expression favor? So does this have some interaction with photosynthesis or maybe with stomatal opening?
Well, LHY is very responsive to light and to temperature, and this is why sort of the proxy for the oscillator for dawn, and the expression is highest at dawn. And we know that LHY is a transcription factor, but because it's part of the oscillator, it targets in a very specific time of the day. So this is one way to see it. When you have a phase shift in LHY, all the genes, they are dependent on this transcriptional factor. They might be affected. We also know that metabolites, they target the oscillator and regulate it. It might be that there is a metabolite in sugarcane that we don't know that might target LHY somehow, either directly or indirectly, and might have a role in physiological changes in anticipation of LHY. It can be also be environmental, and it can be related to temperature. It can be related to photoperiod. Those planes that were submitted to different photoperiod. It was a slight difference, but it was there. And it is yet to be found how sensitive those grasses can be to such minor, fine changes. So this is what I would speculate about.
Terrific, thank you. So I have a question for Nikki from our good friend Keith Lindsay, who asks, what do you think the mechanism is for differential temperature effects on the anther or style? Sort of like what's the mechanism maybe there between the temperature change and sort of the elongation patterns?
Yeah, so that's a really interesting question. There's not too much information available now. I know that the Hormonal lab is doing transcriptome sequencing on anther-filaments and styles at the moment, and we're also doing some temperature style transcriptomes. So that will hopefully start answering that question. But what we do know is that from previous studies, there was a nice paper in 2000, Labello, where they looked at phytohormone and light control of anther-filaments. And what they found was if you expose the anthers or the flowers to constant light, the suppressed anther-filament elongation completely, so the anthers didn't emerge at all. And when they applied auxin to these plants, then anther-filament elongation was initiated. So there is definitely an interplay between auxin degradation and light control and auxin versus light that's promoting anther-filament elongation. They also found there seems to be an antagonistic relationship with gibberellic acid. So under normal conditions, if they sprayed gibberellic acid, it suppressed anther-filament elongation. So something going on there. I also, for the styles and the temperature links, I kind of like the idea of PIF4 and phytochromes playing a role in cell elongation there. We haven't found any data to back that up yet, but I'm hoping some of our sequences will at some point help us answer that a bit further. Yeah, that's pretty much my answer for now.
Terrific. And I have a question from Rajesh for Louisa. Do you think that light intensity, either low or high, could influence the circadian clock irrespective of wavelength?
Yes, it's a big